This is a DNA strand. The backbone of the molecule is a phosphate attached to a deoxyribose sugar. This is linked to the nitrogenous bases, which connect the backbones. Adenine pairs with thymine, and cytosine pairs with guanine. The first step in transcription is initiation. In this step, RNA polymerase binds a promoter to the DNA. Next is elongation. In this step, RNA polymerase unzips the DNA strand and adds RNA nucleotides to complement one gene of the DNA sequence. Notice how the adenine in DNA pairs with uracil in RNA. Also, the sugar in RNA is ribose, not deoxyribose. The finished product is mRNA. In termination of transcription, the mRNA strand detaches from the DNA when the gene is copied, and the termination signal codon is reached. Then the mRNA leaves the nucleus to go to cytoplasm, but before the RNA can leave the nucleus, it must be processed. This involves removing the non-coding regions of the strand, which are introns, while keeping the coding regions, exons. Once the introns are removed, the RNA can leave the nucleus and begin translation. The RNA has a 5' cap and a 3' poly A tail. In termination of transcription, the mRNA strand detaches from the DNA when the gene is copied. Now it's time for translation. Translation takes place in the cytoplasm and also has 3 steps. In initiation of translation, the mRNA, tRNA, and a ribosome, a larger and a smaller subunit, all come together. tRNA is a molecule bound to an amino acid on one end and an anticodon on the other end. The anticodon is a triplet of nitrogenous bases that complement the codons on mRNA. In elongation, an anticodon binds to the exposed start codon, which is AUG, of the mRNA in the A site. This complementary binding brings the first amino acid of the tRNA, methionine, closer to the other ribosomes, forms a polypeptide bond in between the amino acids. The ribosome then pulls the mRNA to the length of the next codon and adds the complementary codon in the P site, connecting the 2 amino acids of the tRNA. This happens multiple times until the peptide chain is finished and the stop codon is reached. Then the protein disassembles.